Swayam Prabha. Digital India. Educated India.
I feel honored to introduce Dr. Vijayan. He is a chemical engineer and received his BTech in chemical engineering from ACTech Anna University, Chennai. He went on to pursue his master's degree in chemical and biochemical engineering at the University of Maryland, Baltimore County, and his PhD in chemical engineering at Oklahoma State University. He conducted his postdoctoral research at Johns Hopkins University before joining the Department of Biotechnology at IIT Madras in 2010. His research interests are in the area of biomaterials and their applications. He is currently working on the development of bioactive wound dressing materials, fabrication of nanocomposite scaffolds for bone tissue engineering, design of novel self-assembling systems for biomedical applications, and surface modification of polymeric biomaterials. So we're excited to hear what you have for us today.
Thank you. Okay. So let me just check if the slide changer is working, but give me a second. Okay, good morning all. Thank you for the kind introduction. So as we were told, I'm Professor Vijayan. I'm actually a faculty in the Department of Biotechnology. My research interests are in the area of materials, right? So how you use polymeric materials for various biomedical applications. So I look at materials as a way to augment tissue and basically try and restore what tissues can do. Right, so tissues are usually damaged. I did talk to you a little bit about what I do and so on earlier. So tissue is something which can get damaged for various reasons, and thank you. So tissue functionality is lost and tissues are damaged for various reasons. It could be injury, like some accidents where you break your bone or something, or it could be some diseases. The example I have shown there is a foot ulcer, which is commonly seen in diabetic patients, right? So this kind of a foot ulcer can actually cause serious problems in people because they don't heal quickly. And it can also be congenital defects, right? So the image I have shown there has a septal defect, and this is usually the found at birth. Right? Any defect which happens at birth is called congenital defect, and you would have all seen movies where they said there is a hole in the heart, right? So that's what this is, like where you have a septal defect and that leads to complications. So this kind of a situation can actually lead to poor quality of life or even death, depending on how serious the tissue functionality is lost, right? So so if you are looking at a fractured bone, it definitely affects the quality of life. It takes a long time to heal, and even after healing there are always some risks of re-injury, or you can actually have a weaker bone depending on age. There may be other issues and so on. So the goal of my lab is to restore damaged tissues so that they can function as they were originally intended to. So for this I broadly call the 3 R's of tissue restoration, which is the regenerate, repair and replace. So regeneration is basically where we try to provide some support in a way that the cells can, in the way that the tissue can actually grow back into its original condition, right? So that is regeneration. So we have different approaches. So there are 3 main approaches. Like one is the material based approach. A lot of my lab focuses on the material based approach. There is also a cell based approach where you can use stem cells and other cells which could potentially be used for this kind of regeneration. And then you can have a combination approach where the cells can be seeded on the material, and the combination of the cell seeded materials can be used for regeneration. And then you have a repair, which is your delivery of drug, and the last one is replace, where you try to replace the damaged tissues because you are not able to regenerate it or replace it, right? So when it has gone to a point where it absolutely needs replacement, then you look at using synthetic materials or some alternative options which can be used for some type of replacements. So let's first look at regeneration. So there are two domains broadly, like one is called the tissue engineering and other is called regenerative medicine. So I will primarily focus on tissue engineering. There is a slight difference between the two, but these two terms are used interchangeably most of the times. Okay, tissue engineering is where you try to engineer tissues and organs for regeneration, and if you are talking about regenerative medicine, it's about providing a support system inside the body to regenerate it, rather than engineer it in a lab setting. Right? In a tissue engineering system, what you do is you try to develop the tissue or the organ in an in vitro system, which means in a lab setting, and then you try to use that inside the body. Okay, so versus regeneration, regenerative medicine, where it happens inside the body itself. So it's a slight difference, but again, they are used interchangeably. So tissue engineering, the terminology is new. It's not something many of you might have even heard of, right? So you would have heard of genetic engineering. So what is genetic engineering? Modification of genes and so on, right? So tissue engineering is not modification per se, but about making of tissues, right? So you are trying to make new tissues in an artificial setting. So you use different materials, it could be natural or synthetic materials, and try to create a tissue in an in vitro setting. While the term is new, the references or the ideas have existed for a long time. So right, so I'm not talking about these as scientific facts, I'm talking about these as human imagination. Right, the idea of engineering tissue is as old as human imagination. Right, so when people lost their loved ones, or people lost some functionality of a tissue, or lost a limb, right, so the imagination of or visualization of being able to restore this tissue has always existed. So these are some examples. So what you see here are basically some examples where you can see that people have thought of these either for replacing lost tissues or for enhancing functionalities. Right, so all of you must have heard of Lord Ganesha's story, right? So where his human head gets chopped off by Lord Shiva, and the first head they find is the one which they used to replace, and it ends up being an elephant head and so on. So this is again an idea of how to use a replacement of tissue or something like that. Right, and there are other mythological characters like Lord Narasimha or Kamadhenu or Yali. So I have specifically chosen these two, like Kamadhenu and Yali, for a reason. I will talk about it in the next slide. So this imagination is not something which is unique for Hindu mythology. Okay, so I want to be very clear about it, because the similar thoughts have existed with other mythology as well. So just take a look at what Kamadhenu is. Kamadhenu is a female head with the body of a cow, wings of a peacock and so on, right? So that's what a Kamadhenu is. And Yali is basically, and I think an elephant tusks, a lion head, and a body with a tail which is a snake and so on, right? So that's what Yali is. You would have seen these in temples. If you go to some of the older temples, you would see these big statues everywhere. So why I brought these up is, you look at Al-Buraq, which is an Islamic mythological character. So it looks very similar to what Kamadhenu is. The only difference being, instead of cow being the main animal, it's a mule, right? It's basically a half mule, half donkey animal. So the logic being that cows were probably more commonly seen and more revered in places where Hinduism thrived, and in places where Islam thrived, it was the other type of creature, which based on which they have built everything. And it again has a woman's head and a peacock's tail and so on. And then you have Chimera, which is a Greek mythological creature. So the Chimera is what you would see in most of the tissue engineering textbooks, because most of them are written by Western authors, and they would talk about how Chimera is a mythological creature which actually talks about enhancing tissue functionality and so on. So this again looks reasonably similar to what you would see for a yali or a sarabam, right? So you basically have a lion with a snake's tail as the tail as a snake's head and so on, right? And it's just that elephants were probably not very common in this region, so obviously they didn't have elephants as part of it. Right, so and you have other creatures like Tarask, which is part of the Christian mythology and so on. So as long as humans existed, they have always wanted to somehow blend the great features of various living beings to get something which is really cool. Right, so that's the thought process has existed. So now coming back to something which is a little more of recorded history rather than what beliefs are. So there have been folklores of nose transplant as aged as back as 1000 BC. So noses were lost in battles and syphilis, and loss of nose is obviously an aesthetic loss. So for that reason, people try to reconstruct the nose, and they have tried it. Obviously they failed because it was not something which people fully understood. But the first plastic surgery, which is in the recorded history, medical history, was done here in India by a surgeon named Shushruta. So he is considered to be the father of surgery. So you would be able to see his history, and he tried skin grafts for reconstructive surgery. So this is again what plastic surgeons do today. They try to use skin from other parts of the body to reconstruct your damaged parts, right? So in 16th century, there have been attempts of using nose replacements using forearm flap and so on. So transplantation of teeth, cornea and skin have been performed in 1700s and 1800s quite commonly, and obviously none of them were really successful for various reasons. Simple things like blood type matching, tissue type matching, aseptic conditions were all not understood at that time, right? So obviously you would not be able to get a successful transplant. You need to have any surgery would first of all need an aseptic condition, right? Which means there should not be any bacterial contamination. When you didn't even know that bacteria existed, obviously you would not be able to create an environment where bacteria was not present. Okay, so an important breakthrough happened in 1933 where tumor cells were wrapped in a polymer and implanted into a pig to show that it was not getting rejected. Because see, whenever you place a foreign tissue in an animal's body, in any animal's body, it will get rejected because our active immune system will go and attack it and try to reject the material. So that didn't happen when it was covered with a polymer, showing that you can actually create some immunoprotection. Kidney transplant was attempted in 1936, however it was unsuccessful, and then liver and bone marrow transplants were tried in 1960s. Lung transplant was tried in 1963 and heart transplant was done in 1967. So modern era of tissue engineering itself started only in the 1980s. So whatever I am talking about before that are all transplants, right? So where you are taking somebody else's organ or tissue and trying to replace the damaged tissue or organ. So that is more of replacement using natural means, not really engineering of tissues. Tissue engineering itself was probably from the 1980s. So we talked about chimera, right? We showed what chimera is. So I'll show you what a modern day chimera is. This was developed by Charles Vacanti in 1997. This is what it looks like. So this is a skid mice or an immunodeficient mice on which they placed a polymer scaffold and seeded it with cow knee chondrocytes. So chondrocytes are basically cells which are present in the cartilage, and these cells were used to seed. So any of your ear is made of cartilage, right? So the cells which are there is chondrocytes. So you try to grow it like this. Obviously this won't be a functional ear, right? It will not have the auditory functions of a ear, but of an ear, but it will give you the plastic surgery effect where you can actually look at it for aesthetic reasons and so on. So this actually was developed in 1997 and was reported in scientific journals. So in the late 90s, when email forwards were the way misinformation was spread. This was something which was forwarded all over the place, and many of us thought that this was some doctored image, but it's not. It's actually some scientific finding which was developed. So this is an important development because you can show that when you can regulate the immune system, you will be able to actually engineer organs in another animal's body, right? Which means you can potentially use it as a farm where you can grow these organs. So moving to more recent studies, this is a surgery which was performed in China, where the person actually had a nose which was grown on his forehead. So this person lost his nose in an accident. So for him to get a nose replacement, instead of just doing reconstructive surgery, they placed a polymer scaffold on his forehead, allowed it to grow with his own cells. It's obviously his own cells will migrate into this polymer and grow, and it formed a natural nose, and this was used to replace his damaged nose. So again, it will not have the olfactory functions, but it will serve the aesthetic purpose. So the ear in a forearm, this is again something which has been shown. So if you search for an ear on forearm on Google, you will see one crazy guy having a ear on his forearm. So he just wanted to have a ear on his forearm for no good reason actually. So he did that, but this has also been used in surgical practices for defense personnel. Right? When people have lost their ears in wars, this strategy has been used where people have been able to grow a ear on their forearm and used it for replacement. Okay, so now that I have told you some history and what people have been doing, let's try and define what this field is. What is tissue engineering? So this is the definition which was published in 1993. So this is a very all-encompassing definition, right? So it says an interdisciplinary field that applies the principles of engineering and life sciences towards the development of biological substitutes that restore, maintain or improve tissue function or a whole organ. So this is a very broad definition. So basically it says that you bring in multiple fields together and try to make sure that the tissue gets back to its original form. So this is the broad idea of how you do it. So there is a tissue which is not working properly. So what you do is you take cells from this person, you then grow these cells, you get enough of these cells, you put them on a scaffold, you put some signals along with this material, and you will get the tissue. And once you have this tissue, you can put it back in. Okay, so this was defined in 1993. So at that time, this was done by two main people who were involved in this were Robert Langer, who is a chemical engineer. So this data is probably a little old. He probably has more publications and patents by now. So he basically is somebody who has hundreds of papers and hundreds of patents, and a lot of it has also been commercialized. Some of you might have heard of the Moderna vaccine. Have you heard of the Moderna vaccine? Yes? What vaccine was that? COVID vaccine, yeah. So that's one of the COVID vaccines which was available in the US. It was not available in India. So there were some RNA vaccines which were made available in the US, right? So Pfizer was one and Moderna was the other RNA vaccine. So he is one of the founding directors of that company and is involved in the development of that vaccine as well. So his work is on delivery of molecules. So he's worked on many things, and so he's been one of the most successful researchers when it comes to translating the research into the field, into the market. So another person who was involved was a surgeon, so who's Joseph Vacanti. Again, he's also a renowned surgeon with a lot of research experience. So these two people together were the ones who published the journal article in 1993. But at that time, people believed that by the turn of the century, which is by year 2000, we should be able to have a fully functional beating heart on a petri dish. That was the thought process people had, because the logic seemed very simple, right? You take cells, you grow them, you put them with some material, and you give them signals, and you will get the tissue. So it seems very, very simple, but it is not. So it has been more than 30 years, and we still don't have a beating heart on a petri dish. All we have is a small beating patch of tissue, heart tissue, which was like developed 5-6 years back, where a small patch of tissue which could beat was developed. So tissue engineering broadly has 3 main arms. So if you look at, so I talked about the general process of tissue engineering, right? How do you accomplish that? First thing you do is take cells. Cells are parts of tissue, right? So anytime we talk about tissue, the first definition which is given to a student is a bunch of cells, a group of cells form a tissue, right? So a group of cells by themselves do not form a tissue. It's not like I can put some cells together and it will become a tissue. A tissue has a lot more components to it. So if you have let's say you have a bunch of cells, right? So you put all cells together and you form like one big blob of cells. Like, that's not a tissue. Actually, those cells can't even survive. Why? Why won't the cells survive? It will not get nutrition, right? So how do cells get nutrition? Extracellular matrix provides some signals, not nutrition. Nutrition comes from? Sir, speak up please. I'm not able to hear you. You said something. So transported by the blood. So basically blood brings the nutrients in. Right? So which means there has to be blood vessels. Right? So blood vessels have to be formed in a certain way. You have to have that, only then the cells will get the nutrition. And also the toxins which are generated has to be removed. That again happens through the blood vessels. So without blood vessels, you will not have a functional tissue, even a surviving tissue. And then you have nerves, right? So you have to have a nerve structure. So only then you will have any sensation or any functionality from the tissue. So for having all that, you need to have like various things. So extracellular matrix is something again which is there for the cells to even attach and grow. Otherwise, it's just a blob of cells and these cells will die. Right? So it cannot proliferate, it cannot migrate, none of it can happen unless the cells can actually function the way they are supposed to. Right? So for that reason, you need these components. So now cells, identifying the cells is the first step. So the next step is to identify scaffolds. So these scaffolds need to have specific properties. They should be able to, first and foremost, they should not kill the cells. Right? I'm using a material. If I put cells on it, the cells should not die. The cell should like the material, which means they should be okay with attaching on the surface of the material and actually growing. Right? So that is important. So and beyond that, there are many functionalities. It should be able to allow cells to grow inside. It should be able to allow blood vessels to form. It should be able to provide the mechanical support. See, the scaffold which I use for a bone tissue versus a scaffold I use for a skin tissue cannot be the same. Right? A bone is much harder. So it needs to have different mechanical properties. It needs to be able to support different functionalities. So depending on that, you would actually have to do different things. And then there are signals. Right, so signals are very critical. There are different types of signals. There are chemical signals, chemical molecules and biological molecules which provide signals. So growth factors are something you might have heard of. So have you studied about growth factors? Yes. So growth factors are molecules which will provide certain signals. It will suggest the cells to behave a certain way. So there are different types of growth factors which can provide signals. And then you also have physical signals. Right, so there are signals which are given through electrical stimuli, and there are signals given through mechanical stimuli. Right? So if I am standing and walking, my bone is getting mechanically stimulated. Right? So the bone formation and bone remodeling happens because of these mechanical stimuli. Without mechanical stimuli, the bone will not be strong. Right? So the muscular atrophy is something we have seen with people, right? So if your muscles are not used, so I don't know if any of you have been unfortunate to suffer a fracture in your life, but if you have, you would have seen that you would have kept your muscle or your tissue not moving for a long time, and then it's so damn hard to move your body, right? Because the muscles have kind of frozen. They are not mobile enough or like flexible enough. You have had that experience? Okay, what happened with you?
Okay, I'm
Okay.
So I had to be in a cast for 4 weeks.
Full.
And then after that, once it was removed, I felt like that ankle was not even there. It took me very long to again mobilize that.
Yeah. So it takes a lot of physiotherapy and a lot of work to get that going. So I had a dislocated shoulder and I couldn't move my hand beyond this point. 4 weeks of immobilization, only about this. Like if I lift my hand, it like I was like a Hail Hitler kind of movement. That's all I could do. I couldn't do anything beyond that. It was bad. So it takes a long time to get your body moving because your tissue gets remodeled with all these kinds of signals. When there is no signal, your tissue becomes kind of dead. So so let's look at, so these are the three main things. So now each of these can be used as approaches to actually restore tissues. So let's look at scaffold based approach, which is where you try to use the material as the base and you try to regenerate the tissue. So there are various types of materials which you can use. So biomaterials are broadly classified as metals, ceramics, carbons, polymers and composites. So these are the 5 major classifications. So actually the 4 are unique classifications. Combinations is basically a mix of these, right? So if you metals are used for various implants. So only now metals are being explored for regenerative medicine or tissue engineering, but they have always been used for implants. Yeah. Scaffold is? Yes, the scaffold is a material on which the cells can grow. So it's a substitute for the extracellular matrix. Right? So your extracellular matrix provides the environment for the cells to attach and grow. So the scaffold will behave that way. Okay. So metals are generally used as implants. So it's used for replacements rather than being used for tissue restoration. But recently there has been growing interest in using magnesium alloys for being used as scaffolds for bone tissue engineering. So then you have ceramics. Ceramics, some examples are alumina, zirconia, hydroxyapatite and various things, tricalcium phosphate. These are some examples of ceramics. So you would have seen ceramics in your life, right? So ceramics are very commonly seen as porcelain at home. So where do you think ceramic would be useful in human body? Bones and teeth are all made of ceramics, right? So it has a lot of ceramic to it. So I'm sure at least 1 or 2 of you would have had a root canal done already. Yes. Yeah. So the odds are like 1 every 50 at the age of 16 have gone through root canal. So it's crazy. But anyways, so root canal, after doing root canal, you would have had a crown in some cases. If you keep a crown on top of your teeth, you will be given an option of choosing a metallic crown or a ceramic crown. Right? The ceramic one looks similar to what your natural teeth is. Right? So ceramic is basically what is present in your bone and teeth, and it is commonly used for bone tissue engineering applications. Hydroxyapatite is actually the material which is present in your bone. Okay, so this is present along with other components, but the ceramic component is hydroxyapatite. Then you have carbons. Carbons like carbon nanotubes, graphene oxide, all these have become of interest in the recent past. So they are not naturally present in your body. Carbon is present as organic components, but carbon by itself is not present. It's not like you have graphite and graphene and diamond in your body. Right? So those things are not present in your body in that form. So they usually present as some organic compound. But there is a lot of interest in using these materials as well, where people try to use carbons and carbon nanotubes or graphene because they have some desirable properties. Some of them are useful for improving blood vessel formation. Some of them have antibacterial properties. They can also have electrical conductivity. Right? So the material can have some electrical conductivity. Right? So when the material is electrically conductive, it can be used for growing different types of tissues. Okay. So for example, a neural tissue. The neural tissue needs to conduct electricity. The cells will conduct electricity. So providing a base which will conduct electricity will help in alignment of the cells and so on. Right, so lot of interest has been shown in carbons, but before being used for tissue engineering, carbons were used for coating. So there are types of carbons like DLC, diamond-like carbon, which is used for coating on heart valves and other blood contact materials, because these carbons have very good compatibility with blood. So when blood comes in contact with a foreign material, the blood will start reacting and it will start forming a clot. Right, so if you put some implant and it comes in contact with blood, there will be a blood clot which is formed. Blood clot can be a big problem because this blood clot will occlude the pathway of blood, and it can actually get detached and it will go and block the blood flow in some other region as well. So it can result in heart attacks or stroke and so on. So you do not want that. So for that reason, they use diamond-like carbon and other carbon coatings on heart valves to improve the blood compatibility. Polymers are the most extensively studied class of materials for tissue regeneration, because polymer, a lot of components present on our body are polymers. So there are different proteins, there are glycosaminoglycans, there are various other carbohydrate polymers. Everything in our body is some type of polymer, right? So you have a lot of polymers which have specific properties which can actually serve the purpose of an extracellular matrix. So we do study a lot about polymers. And composites are basically combinations of these. So people have tried polymers along with carbons, polymers with ceramics and metals, and various combinations to achieve specific properties. So that's a composite. Then you have the cell-based approach. So with cell-based approach, there are different types. So there is autologous cells, allogenic cells, and xenogenic cells. Autologous cells are cells which are taken from your own body. When you take cells from your own body to replace or regenerate some tissue, it is called autologous transplant. So can you give me an example for an autologous transplant? Skin graft. Right, so in case of plastic surgery, skin grafts are used where you take skin from another part of the body. In burn injuries and so on, it's very commonly done. Any other example?
So when the spine is damaged, they take bones from the tires to try to...
Bone grafts. So bone grafts can be used. So you can actually take bone pieces from non-load bearing bones. Okay, so bones have, there are two types of bones. You have load bearing bones and non-load bearing bones. So non-load bearing bones are of lesser importance than load bearing bones when it comes to your stability and your mobility and so on. So for that reason, you can actually take some fragment from the non-load bearing bone to heal a load bearing bone. That is one option. What else? Head, hair transplant. Yeah, potentially hair transplants can be done. Yes. So something a little more invasive. Because heart transplant, the chance of immune rejection are very low. Yes, bypass surgeries. So in case of bypass surgeries, what you do is you take a vein or a blood vessel, usually from the calf, and you use it for bypassing a blockage in your heart. Right. So how many of you know what a bypass surgery is? Okay. So for others, a bypass surgery is when your blood vessel is blocked because of some, it's called atherosclerosis. It's basically some plaque formation. If it blocks the blood vessel, blood flow, then it can cause a lot of stress to your heart and result in a heart attack, which will damage your heart and you will, it can lead to death. So for that reason, this blockage can be bypassed by attaching a new tube to it. So this is like building a bypass road to avoid city congestion, right? So you build a road outside which you can take if you don't want to go through the congestion. So you create a bypass to avoid this blockage. So that is so those are places where autologous transplants are done. What about allogenic? So can you give me examples of allogenic transplants? Liver transplants, kidney transplant, heart transplant, various such transplants where you usually take a full organ from another person and use it for your body, or corneal transplant. Various things like this have been done, and those are allogenic transplants. And then you have xenogenic transplants. So in allogenic transplants, you are getting it from another human being, right? A person of the same species. Xenogenic is where you can take it from a different species. Can you give me an example? Can you think of a place where you can take a tissue from some other organism and use it in your body? It's being used surgically currently. Sorry? Pig heart valve is used, not the entire heart, the heart valve is used. Pig's heart valve is actually something which has been used, which is always used usually for older patients. So mechanical heart valves are used for younger patients, and your pig's heart valves are used for older patients. Okay, so those are some places where you have used different xenogenic transplants for restoring the tissue functionality. So another place where a lot of research is happening is trying to use pig's pancreatic tissue. Like your pig's insulin is very similar to human insulin, and you have functionality is also very similar. So a lot of people are working on using pig's pancreas for treating diabetes, right? So the advantage of autologous transplant is there is no risk of immune rejection because it's your own cells or your own organ, and there is no disease transmission either because it's within yourself. In allogenic, there is the advantages is less expensive, and it's more available. Right? In case of autologous, there is only limited availability. There is only so much of tissue in your body, and usually if the patient is already suffering, there is not enough tissue to be harvested from the patient. So getting somebody from someone else makes the availability better, but again there is some risk of immune rejection and disease transmission. However, in case of xenogenic, it's easily available, it's least expensive, but there is a very high risk of rejection and disease transmission. So amongst the cell-based approach, there is also different types of cells that can be used. You could either use differentiated cells which match this tissue type, or you can use stem cells which do not have the functionality but are more abundantly available, right? So more easily growable. Right, so basically, if you have a fully differentiated tissue cell, this cell will not grow very easily. The cell will actually take a lot of time to grow. So it's kind of difficult to culture these cells, and there will also be some donor site morbidity, which means that the donor site will be damaged, and that will take some time to heal. However, if you are using stem cells, you do not have these limitations, but the problem is it can lack functionality. Because stem cells are basically cells which are growing and they are not differentiated, so they don't have any functionality. But the advantage is they are more easily available. They can be cultured quickly because they grow very quickly. So it's quite advantageous that way. But the problem is they can also have uncontrolled growth. Right, so it can lead to something called teratoma formation. It's type of a cancer. It can form some kind of a tumor. So that's also a risk which is associated with stem cells, especially something like an embryonic stem cell. So teratoma formation is an issue. So there are different types of stem cells themselves. There is something called adult stem cells, which is from adults, which is taken from adults, and then you have fetal stem cells, which is from a fetus, and you have embryonic stem cells, which is isolated from an embryo. So it's from a blastocyst of an embryo, you can take it. And then you have iPSCs or induced pluripotent stem cells, where you basically take an adult differentiated cell and you de-differentiate it to form a pluripotent stem cell. So there are some genes which you can insert to make them into induced pluripotent stem cells. So the invention of iPSCs happened, I think, in 2006, if I'm right, like around that time maybe. And 2012, they won a Nobel Prize for that discovery. Okay. So these are some cell-based approaches. Then we will talk about the signal-based approach. So signal-based approach usually means that, yeah, please. So, yeah. So donor site morbidity is when you are taking cells or tissue from one particular source, that you are damaging that source, right? So let's say you are talking about a patient who has gone through some burn injury, and you have to use a skin graft. So let's say you remove skin from the thigh, right? Now you are causing some injury to the thigh, and that needs to heal. So the patient is already suffering, so you are causing more injury to them. So which means that tissue, it might take longer for that body part tissue to heal. So that can lead to morbidity of that tissue. So that is called donor site morbidity. Okay. So same question. Okay. So in the signal based approach, we try to deliver the signaling molecules. So here is an example of a commercially available material called Infuse, which is a bone graft developed by a company called Medtronic. So they have they are delivering a molecule called BMP, which is bone morphogenetic protein. So this is a recombinant version of this bone morphogenetic protein. So this helps in bone formation. So they load it to a sponge, a collagen sponge, and this particular growth factor gets released, and due to the release of this growth factor, there is better bone formation. Okay. So that kind of covers the various aspects of tissue engineering, a broad aspect. I have not gone into the details of any of the things. So if you are interested, you can watch more detailed video lectures. So then I have a course on tissue engineering on NPTEL. So if you go to, how many of you know what NPTEL is? Okay, a good chunk of you seem to know. So if you don't, please go and search for NPTEL. So it's National Program on Technology Enhanced Learning. So this is a Government of India supported project where we have multiple courses. There are almost 700 courses are offered every semester. So where students can actually register for free, and so enroll for free and start doing watching videos and submitting assignments. If you want a certificate, you can then go and apply for an exam. So the exam fee is like 1,000 rupees. You can apply for the exam, appear for the exam, and if you score at least 50% in the exam, you will be given a certificate from the corresponding IIT. So NPTEL certificate has a lot of value. A lot of school students do take these exams. So I don't know if have any of you taken an NPTEL exam before? No. Okay. So if you are interested, you can do that. So this is quite, there are many interesting topics there. So one of the courses there is my course on tissue engineering. So if you are interested in to know more about tissue engineering, you can go and watch that. So the videos anyways, you don't have to pay anything. You don't have to even enroll. You can, it's on NPTEL website, they are available for free to watch. So if you are interested in getting a certification and so on, you can go and enroll and register for the exams. Okay. So there are many other courses on NPTEL as well. So you can, I would strongly encourage you to go and do that, especially if you are interested in learning new languages. There are really nice courses on German and so on. I think the course on German is recognized as some equivalence to some German certification and so on. So you will have your own, you will be able to find the courses which fit your interests on NPTEL. Okay, moving on to the next aspect, which is, sorry, bone morphogenetic protein.
So is there any part of the body where that's not being used?
Okay. So that's an interesting question. Let's discuss that a little bit. So the question is, are there any parts of the body where we can't directly use this type of a method? Brain, okay. Heart, okay. Any vital organs? Eyes, okay. So intestine, okay. So why? Okay, now let's. Blood, okay. Kidneys, okay. So now we have like different organs which you have named. Let's try and look at why you thought that was an organ which you couldn't use. So people who said brain, why? Okay, so brain is highly sensitive. You would not be able to do some changes to it and expect it to function properly. Okay, or heart. Somebody said heart.
Very important for life, and if something goes wrong, let's say there is factor that we have to take note as well, right?
Okay.
If that happens, then happens in the heart. That's not something really won't happen.
Okay. So being an essential organ, like highly important thing which can immediately affect the life, so that's an organ you feel you cannot do it. What about others, like you said eyes? Sorry.
What about like
the nose itself cannot perform the olfactory functions. The heart has various neural functions, so it's not functioning that well.
Okay, so the question is, so what she's saying is, you said that nose the people are doing, but it doesn't have olfactory functions. You said ears are there, they don't have auditory functions, and what's the point of having an organ which doesn't function? Fair enough. So you said eyes, right?
I said blood.
Blood, sorry. Why do you think blood would be difficult?
Sir, because it's quite dynamic, it keeps flowing throughout the body, and any change that we make would kind of affect all the parts because it is.
Okay, so there is going to be an interconnection and so on, right? So you said kidney.
So technically kidney is not a vital organ, but kidney is directly connected to some of the main blood vessels in our body. Like he said, probably the
Okay, so yeah.
Also, blood has a lot of proteins, so it would be kind of difficult to identify each and every protein and kind of
Okay.
match.
Okay, so this is an interesting point of discussion, right? So because now two things you have to look at. So when I talked about examples, I didn't specify why I am using these examples, but I kind of talked about a few specific examples, right? I talked about bone, I talked about cartilage, I talked about skin. What is common amongst these?
They don't respond to stimuli.
They do respond to stimuli. There is a they do respond to stimuli. They can repair by themselves. Partly true for it's true for bone and skin, not for cartilage. Cartilage cannot repair itself. Voluntary movement. Okay. They are connective tissues and connected tissues. Okay. Sorry. Not able to. Comparatively abundant. Okay, abundance is not a factor here. So, okay, let me give you the answer. So the thing is, these are all like structural components of your body. They do not have any biological functions. They provide mechanical support, they provide structural integrity, they provide basic protection and so on. So skin, yes, it does have certain functionality like perspiration and so on. So the engineered skin cannot have all that. So it is only to provide basic function. Sorry, bone marrow of the bone is a different thing. I'm talking only about, see, when you're talking about replacing of bone, you're only looking at the bone, not the bone marrow. Bone marrow is a completely different thing, so you cannot that's bone marrow is not a function of the bone, right? So bone marrow is a separate tissue by itself. Okay, so the reason we use these is these are not functional organs. When you are talking about functional organs, it brings in more difficult challenges. So actually heart is a lesser challenge because its functionality is still only a mechanical function, primarily mechanical and electrical, right? It does not have any biological responses. An organ which is much harder to engineer would be a pancreas. So why would a pancreas be hard to design? It secretes various things, right? And based on a feedback mechanism. So it has to sense that there is some glucose and actually release insulin. If there is going to be lesser glucose, then it has to respond accordingly. So it has a very intricate mechanism. And if I am going to have some other tissue, it also should be able to allow glucose to pass inside, and it should be able to sense the glucose, and it should be able to allow insulin and other proteins to come out. But at the same time, it cannot allow immunoglobulins or interleukins to come in, because then it will have an immune reaction. So it's like a very complicated system. So anytime you have a biologically functional tissue, it becomes much harder to develop. But yes, the same principles can be applied, but how you apply the principles will change. So the type of material you use and what will be the cells you use, how complex does the system have to be, all that will change. So one of the points which was brought out when we talked about blood is, you said that blood has too many components and it seems like a very complex fluid to work with. So that brings in the point of how good is good enough, right? So do you want to make it perfect, or do you want to make a tissue which will do its job? all we need to do is it should do its job. If we want to make a perfect tissue, that's never going to happen. Like we are never going to be able to match the natural tissue, right? So think about what our heart does. A heart beats from the day you are born till the day we die, right? So how many times is that? Roughly 72 times a minute. So how many times a day? That's what I think 1440 minutes a day. So that into 72, that many number of times a day. So if you're talking about at least 10,000 times a day, roughly, no, 100,000 times a day, roughly 100,000 times a day. And let's say a person lives for like 70 years, that's roughly around 28,000 days. You're talking about 28,000 into 100,000 times the heart has to beat. No, and what does a heartbeat mean? There are valves inside which are opening and closing for every beat, right? So this valve, compare that with what you have for your taps. That's also a valve. Can you try doing it 100,000 times and expect the valve to function? Chanceless, right? So that's not going to happen. So even the mechanical heart valves that we have developed, after years and years of research, we have developed very successful mechanical heart valves. They have a life of about 10 to 15 years. After 15 years, anything which comes is bonus. You do not have any chance beyond 15 years, right? So that's the way it is. So, but natural heart can function for 70 years easily. Even when you abuse it, right? And we treat it so poorly with all the dietary habits we have and the sedentary lifestyle we lead, we treat heart so poorly and it can still function for such a long time. Right? So we will never be able to match what the natural tissue is. That's not the goal. The goal is to provide something which will solve the problem for as long as possible, and hope that the natural tissue can somehow take over. If you are replacing an entire organ, that's not going to happen. So that's why we try to replace bits and pieces, because then the natural tissue might be able to take over. So if you are talking about bone regeneration, so let's say you lose a bone, a part of the bone, then what you try to do is like whatever infused bone graft or something, you put a graft there. Then what will happen is this graft is going to help support the bone growth for a period of time. So maybe for 4 months it will provide some support, or 6 months it will provide some support. By then we hope that the natural bone will form and this natural bone will take over, right? And when this healing happens, what we try to do by providing these kinds of grafts and supports is to make sure that the newly formed bone or the healed bone is as good as the old bone. Because otherwise, if it heals by itself, it might not have that kind of a healing. It might actually have some kind of hindered healing. So if you look at how the tissue looks, a bone tissue, when you